So I mentioned to you briefly this idea of sex chromosomes when we talked about Fragile X. Now, when we look at Fragile X, we'll talk about it more in evolution and think about its effect on males and the idea that having a single copy of that X chromosome is detrimental because they only have one X compared to females. We can look at that sex-linked trait being carried through to other characteristics as well. So the idea of this inheritance of chromosomes and the idea that we inherit specific alleles on our chromosomes from mom and dad, this independent assortment idea we've already talked about, right? So what I want to focus on now is less this idea of independent assortment, because we've already talked about it. I give you the definitions here again in case you don't remember. But when we look at these laws of independent assortment and segregation, what we're really interested in is, what do my gametes look like? Because I need to know what chromosomes are in the gametes so that I know what the offspring have a potential of inheriting. So we can go through all kinds of diagrams to show this to you. We've already talked about this in excess when we looked at the idea of a 9-3-3-1 formation in a dihybrid cross. So that gave us variability. That variability we said was extremely important because it gives us the potential eventually for evolution and variable populations. This is really, really important for us. But what we want to know is, how can we keep track of all of this? Well, scientists have developed things known as genetic maps. So just like you have a map of a town or a state, we can actually map your chromosomes and understand how they work together and how they actually link information together. So they started thinking about, of course, plants. I know many of you are tired of talking about plants, but what we think about in this process is, I'm not so much this phenotype stuff anymore. I'm interested in the genotype. I want to know what's going on that we have this predicted 9-3-3-1 ratio, roughly, of offspring every time we cross two heterozygotes. So looking at the genes, what we actually can think about is where this is placed on the chromosomes and what this actually means for our offspring. So if we have what we call linked genes, so this means that if you have a purple flower, what I want to know is, does being a purple flower make you more likely to have long pollen than short pollen? So is there some connection between being a purple flower and having long pollen? Okay? And conversely, in this case, am I going to have a red flower that always has round pollen? Now, you might say, okay, well, if I'm a red flower and that means I have round pollen, that means that these individuals that are red with long pollen can't actually exist. So that's what I want to think about. I want to think about the inheritance of these chromosomes, the inheritance of these alleles. And that's what science works on now in genetics, is trying to understand where these genes lie on our chromosomes. Are they close together? Are they far apart relatively? Okay? And if they take up one of these characteristics, what's the possibility that they're actually going to cross over? Now, for those of you that were with us for our discussion of meiosis in previous lectures, crossing over occurs in prophase 1. of meiosis 1. So this is happening immediately. And what crossing over means is that we can look at the exchange of genetic material between non-sister chromatids. So this is demonstrating crossing over to you. We've got this pair of replicated chromosomes known as a tetrad. And essentially what happens is the legs of these chromosomes, they're literally called legs, the legs of these chromosomes end up intertwining, and you see that I now have, instead of a nice one red chromosome, one blue chromosome as I did before, I now have this mixing of colors. Well, the mixing of colors represents the mixing of genes, or the phenomenon known as crossing over. Why is this important? Well, it means that these linked genes are not actually inherited together all of the time. It means that we get increased variation in our offspring. This is a very good thing. Because when we start actually looking at organisms instead of just flowers, if we actually look at animals, we can look at different body types. We can look at different characteristics. We're looking at phenotypes here because it's easier. And we're going to link those phenotypes then to genotypes. And I want to know if I cross this gray-bodied, long-winged female with this black-bodied, stumpy-winged male, vestigial means, in this case, non-functional. It's a leftover. So this male wouldn't actually fly, okay, because of those stumpy little wings. If this is the case, that black body and vestigial wings are linked, that means that all my individuals with a black body should always inherit vestigial wings. Is that true? Well, no. I've got this gray vestigial thing in the middle here. So that means that this is not holding. Why isn't this holding? They're linked. They're on the same chromosome. Why isn't this holding? Well, it's not holding because I'm getting crossing over. I'm getting increased variation of these alleles and these genes, and this is a critical part to lead us to higher levels of evolution and changes in that regard. They can do this with all kinds of things. So this genetic link map, or what we call distant maps, can show us how close things are together on a chromosome. If they're really close together, they are more likely, so if alleles or genes, alleles are just variations of genes, right? So if genes are close together on the chromosome, they are more likely to be inherited together. Why? Why? Well, what we have to think about is the idea that if you have genes that are close together, it's harder to get crossing over to split them up. If they're further apart, if I'm looking at the idea of these cinnabar eyes being linked to brown eyes, okay, what are the chances of these things kind of matching up with gray body here, okay, red eyes, brown eyes? How am I going to match all this up? Well, these are actually the wild types on the bottom. So how close together are these genes? Wild type means, we'll say normal for now. Okay? So how close are these together? The further apart they are, the more likely they are to get split up by crossing over. We'll talk about that more as we go. So the other thing we have to consider is this idea of sex-linked characteristics, meaning that you have a characteristic that you're inheriting on the sex chromosomes. Now, usually we look at the X chromosome for sex-linked genes. Why? No offense, guys, but the Y chromosome is very, very small. Really, the Y chromosome is carrying information in mammals just for the development of testes. Maleness. In the absence of that Y, there are no testes, so ovaries will develop. The X chromosome, on the other hand, is huge. It carries all kinds of different information. Now, for males, in mammals, we look at XY. In females, we look at XX. There can be variations on this. In other species, they don't have an XY system, okay? So in this case, 2 X's in a grasshopper actually gives you a female, like we would predict, but they have no Y. So males in the grasshoppers and other insects are just X. So a single X. They put an O here as a place filler for you, but really the females are XX, males are just X. Okay? You can look at other variability. There's ZW possibilities in particular species. I'm not particularly concerned about this. I just want you to know that there are other options other than just being XX and XY all the time. Okay? Some actually lack sex chromosomes altogether. In bees, sex is actually determined by chromosome numbers. So we've said before that diploid is 2N, haploid is 1N. The males are haploid, the females are diploid in bees, and a number of other insects as well. So different variations, but what I want to think about is how these sex chromosomes can be affected by the environment and by other chromosomes. So in the environment, we can actually look at sex determination by temperature. And so we can actually look at particular temperatures in nature to determine whether or not the eggs will be male or female. We're worried about this because as global temperatures increase, we're actually seeing a shift in the ratio of males to females. We've talked about this before as being a problem with mate selection, and we'll talk about it more when we look at ecosystems. The sex-linked genes, though, that we look at in mammals and particularly in humans, we can think about X-linked and Y-linked genes. So I mentioned that there isn't a lot going on the Y chromosome, but when there is, it's big. So we've got to think about Y-linked being passed only to a son from a father. Why? Well, if a male is XY and we split up that to produce gametes, Y one direction, X another direction, mom is XX, right? So if we set up our Punnett square, mom is XX, dad is XY, our offspring are XX, XY, XX, XY, Y. So when we look at this, we have the possibility of 2 females or 2 males. The only place the Y can come from and go to is from dad to sons. The X's, on the other hand, could come from mom or could come from dad. So we look at the X-linked genes being passed on from mom both to the son or daughter, because that's all she has to contribute, right? It doesn't matter if she's having a son or a daughter. All she's given up is an X, right? From the X from the father, though, would go to a daughter, okay, based on our Punnett square. So in some species, we look at this being an indicator of particular phenotypes. So fruit flies actually carry eye color on their X chromosome. So again, kind of fruit flies as a poster child for genetics, we can look at eye color as an indicator of whether or not particular genes have been inherited. So we can look at specific traits inherited in fruit flies, and we can track it externally based on eye color. This is certainly easier than cutting open a fruit fly and trying to determine what its genes are. So this link to particular X characteristics based on eye color in the fruit fly has been really important for genetics. Now, we can look at different characteristics that give these offspring particular eye colors associated with them. I'm not going to focus on that for now, but if you're interested in it, it's here for you to consider, and how you would get different combinations of offspring and different colored eye combinations based on whether or not you had the male with a particular eye color, R being the dominant red, or whether or not you had little r with the white eyes, and how that can be inherited and where that information is coming and going. But if we look at X-linked disorders in humans, one of the primary ones that we often associate is colorblindness. Ladies, this does not mean that you cannot be colorblind and you cannot have daughters that are colorblind. If it's carried on the X chromosome, we can certainly look at that X chromosome with color blindness being inherited by either a daughter or a son. From mom. If dad is colorblind, right, that X only goes to whom? Right, his daughter. Now, the nice thing with X-linked disorders is that you often need 2 copies in females to show this, and that's why we see very few females that are colorblind. Because you end up with 1 colorblind X and 1 normal X. Well, as long as you have 1 normal X, typically you have normal color vision. But in males, if you inherit a colorblind X and you have a Y, you only have 1 X, so you would be, in this case, a colorblind male. We have other disorders. We've talked about Fragile X before, more common in males, again, because they're only inheriting 1 X instead of 2 like the females. Hemophilia is another one that we see associated with the X chromosome. So there's a lot going on this X chromosome. Things like muscular dystrophy and so on. There have been stories through history trying to understand these X-linked chromosomes. Again, here setting up a pedigree. We talked about these before, to track family histories, and in this case, looking at the royal family of Russia, and looking at the idea that the colored in squares and circles, remember circles female, squares male, the colored in ones would have expressed the trait of hemophilia. The half colors are just carriers. So remember, because X and X in females, you can be a carrier, in this case, 1 with hemophilia and 1 normal, for many generations. And you see here the P generation, the F1, the F2 generations, all were carriers. And then eventually they had a male son, right? So those carriers, if mom is carrying this hemophilia gene, she's got a 50-50 shot of giving that hemophilia gene to her son, right? So if she gives that to her son, he's going to inherit a hemophilia gene from her and a Y chromosome from dad, meaning that he actually expresses hemophilia. This has led to multiple murders of wives through history, particularly in royal families, before we understood how X-linked diseases were passed on. So the Tsar Nicholas of Russia actually believed that Alexandra had cheated on him and had had an affair because she suddenly had a son with hemophilia, and no one else in the family lineage had ever shown hemophilia. Well, it turns out that they've all been carriers for decades, but finally had shown just by random chance that he got the hemophilia gene, and Tsar Nicholas actually had Alexandra killed. Be careful with your genetics.